Thank you very much. And I realize this is sort of, we're entering happy hour time now, where most people are heading to the pub. So I really appreciate the commitment, and your attendance is very much appreciated. The other thing I want to say is, how much of a pleasure it is to be invited to Keynote back in Norway. Fordi jeg faktisk norsk. And I know that surprises a lot of people with a last name like Letessier. So what I was going to talk about was very much something that's been keeping me engrossed for the last 15 years or so. And specifically the sort of the various signal that we're seeing in pelagic and benthic fish assemblages concerning the influences of MPA, their effectiveness, and the effect of human pressures. A little bit about me. As I mentioned, I'm originally from Norway. I did my PhD in St. Andrews. I did my first postdoc at the University of WA, and I remain an adjunct research fellow there. And then for the last 10 years or so, I've been at the Zoological Society of London. And for the last year, I've been a lecturer in marine conservation at the University of Plymouth. And the bulk of this work was something that we initiated when I was a bit of a visiting scholar at the Center of Synthesis and Analysis for Biodiversity in France. And this broadly is sort of where my interest lies. I'm interested in megafauna, fish, sharks, seabirds, marine mammals, and because their disproportionate effect on ecosystems. And because of that, I tend to care about human pressures and how those things interact in space and time. So I like to say that, space and times are kind of the things I like to play with and tease out signals across these various components. And although I worked a lot in pelagic environments, I also tend not to have any strong sort of taxonomic loyalties. And this is kind of the bulk of the key message really in this paper, which came out about a year ago. In Science, I guess one of the minor highlights was that we got the first page of Le Temps in Switzerland. So we got a landlocked country to care about the oceans, which was great. And the methods that we applied was these things, BRAVs. So these are stereo cameras that we deploy in the midwater and on the seabed. The thrust of this here, you see a midwater system here deployed in French Polynesia. The acronym stands for Baited Remote Underwater Video Systems. And this was the technology initially came from New Zealand, and it was kind of these burly Kiwis who'd come up with the term BRAVs. And then when I started my postdoc on this, we were working on midwater systems. So we came up with the acronym Systems for In-Water Shark and Tuna Analysis, or SISTAs. So we had the BRAVs and the SISTAs, but that one never stuck, unfortunately. And the principle is simple. You have a bait canister. You leave the whole thing to soak on the water, 1 hour on the seabed. This is Blue Abacus. This is my old boss, Jessica Mayerwig, whose team has been spearheading this field initiative. And you leave it to soak for 2 hours in the midwater, 1 hour in the seabed, and then you pull the whole thing up, and then the stereo cameras, which you go through the footage, and you get species ID, relative abundance using something called MaxN, and as well as size, and that really is critical, body size. And in addition to being able to deploy these from a range of vessels, we've taken a considerable amount of effort to calibrate and to validate this method, and to see how it compares to other studies, so UVCs, so scuba surveys. For the pelagics, we were looking at things like echo sounder, and really this gives us a lot of credibility and a lot of power to detect signals in space and time, but also that power comes from the bait. So really, that attracts particular species, large species, carnivores, predators. Here you can see sort of a snapshot of some of the species we're detecting on these bruvs. You have here a great white shark, tiger shark here on the seabed, and you can really see a sort of the diversity of species body sizes that we're getting. And the shark abundance was actually the focus of one of our articles in PLOS Biology. And here we demonstrated that distance to market, so proximity to cities larger than 10,000 people, is actually the most influential driver of shark abundance. So more than all the other environmental and bathymetry signals that you expect that a shark would care about is how close the nearest harbor is. I mean, 10,000 people is typically at that point you have the sort of infrastructure for industrial fleets. So which is why we didn't just arbitrarily choose that size of cities. And at around 12,000 kilometers is where you start seeing sort of pristine levels of shark abundances. And that tells you a lot about the sustainabilities about these fisheries. When we were looking at this particular data set, so shark abundance, we didn't actually see a signal from marine protected areas. But this spatial pattern is actually consistent with population trends. So pelagic species and predators include some of the most vulnerable vertebrates taxa. You can see this here in looking at the sort of the red list index for oceanic sharks and rays over the last 50 years compared to other taxa like mammals, amphibians, seabirds, or just birds, sorry. And this kind of leads us to that question of how much we can reverse this trend through protection intervention. There's been a lot of discussion around pelagic MPAs particularly, and you've heard the argument that pelagic species move around too much for MPAs to be really effective. This is the sampling effort we've done to date with our bruvs. So we have about 10,000 benthic bruvs deployed around the world, 7,000 pelagic bruvs, and this gives us roughly about 20,000 hours of video footage, which has been meticulously analyzed by PhD students, technicians, and this is very much a collaborative effort. The bulk of this data is coming from Australia. Again, that sort of signifies the, I guess, the sort of legacy of where this technology initially was developed. And really, it's a huge collaborative effort. We've been working with National Geographics, with the French Pristine team, with National Geographic, who was also called Pristine Seas, so it was seemed to be quite a popular name. We were working with the Blue Belt, so some of the overseas territories. Myself, I've been doing most of this field work in Chagos, and IMR as well. Evan in the room, who's one of the sort of earliest Norwegians who embraced this technology. And that sort of brings us to our objectives. We wanted to do a global assessment of of pelagic and benthic fish assemblages. Oh, and one thing I should mention, this color scheme here, so for benthic we're going to be looking at orange, and for pelagic blues, that color combination will be the same throughout this talk. Yes, we wanted to conduct an assessment of marine ecosystems in relation to human pressure, and specifically considering competing hypotheses concerning the sensitivity of pelagic and benthic systems and their responses to protection status. And it's from those objectives that we needed to opt for some kind of unifying metric of that allowed us to contrast both pelagic and benthic systems. And this is why we focused on body size. marine ecosystems, and particularly fish assemblages, are beautifully size structured, and which means that you can only eat something that's smaller than yourself. And this is kind of one of the main sort of contrasts between terrestrial and marine ecosystems, really. If I were to tell you that I have a 100-kilo animal in my living room, you wouldn't know if that animal could eat me or I could eat that animal. Whereas in the ocean, it's you know, somewhat simpler. There are exceptions, of course. I'm sure you could think of a few, but that's the sort of, you know, in general, as Peter de Brugge said, you know, the grosse fischen eaten die kleine. Big fish eats the small. And many scientists have made that observation that for every 10-fold increase in body size, you have a 10-fold decrease in abundance or frequency. And if you plot body size against abundance on a logarithmic scale, otherwise known as a size spectra, you get this declining slope of -1. And this relationship holds true for from bacteria to whale. And this was recently confirmed by Hatton and friends. This was looking at the sort of global biomass. And another way of looking at this kind of data is just like a trophic pyramid, but just flipped on its side. You know, so if you just sort of push that 90 degrees, you'd have a trophic pyramid. And that gives us some theoretical expectations with which to compare both you know, empirical observations. That same paper looked at how much this the slope of that pyramid had changed, comparing with pre-industrialized data against the present. And you can see that the present is a lot steeper. So this basically reflects the sort of decline in these larger size classes as a result of whaling, as a result of industrial fishing fleets, etc. And a facet of these size spectra is that when you're actually going in and looking at empirical data, you tend to get these undulations. And there's been a lot of work in the last decade or so trying to explain why do you get these undulations. You know, people are suggesting, well, is this a sort of an artifact of sampling? You know, obviously when you're, you know, the size selectivity of a plankton net, for example, of a bottom trawl, of a longline fisheries, they will all target specific size ranges. So that was one sort of hypothesis. But it seems that the sort of recent research seems to suggest that these are just these consecutive domes are generally thought to represent predator-prey relationships. So it's kind of, you know, you have these peaks, algae, zooplankton, large predator, zooplankton, and prey fish. So not so much an artifact, but actually a signal in functional groups. And a recent contribution was looking at, now we're getting into the theory here, that for pelagic systems, these domes are the result of trophic cascades, so particularly in nutrient-rich waters. So one of their predictions was that at low nutrient levels, size spectra form a straight line, so here. But then as you're starting to get more productivity, starting with the smaller size classes, so phytoplankton, that sort of little peak travels down, and then eventually gets absorbed up in these larger size classes, which then there's no more predation because they basically outgrow predation. And then they sort of lead to a trophic cascade. So then obviously because they're staying here, they integrate across time, then that causes a dip in their prey, and then that sort of travels back down again. So you can imagine this as a kind of a, you know, as the string on a harp just being plucked and then sort of traveling down, reverberating up. Usually I like to have, when I talk to this with my students, I like to have a bit of cable or something that I sort of tend to demonstrate. But and in contrast, on benthic size spectra, you have this large sort of refuge caused by habitat complexity. So imagine that, you know, you have a stagord coral or a kelp forest. It's particularly size classes which will be able to hide in there, and therefore not being able to be eaten as much as, you know, if it was out in the open. So you have that kind of anchoring point. So with that preamble over, this here is the result of our 20,000 BRUVs body sizes. So this is this is 1 million fish that's been measured individually. All the orange one are the seabed ones, so the benthic fish, and the blue ones are the pelagic. So I know it's quite a busy plot, there's quite a lot going on. The first observation is that there's more benthic individuals than pelagic. That might not be a big surprise, but you know, if you want to go fishing, you fish near the seabed, right? There's more likely to be stuff there. The other thing is that for the large ones, you have both benthic and pelagic individuals, but the majority are pelagic. One thing I should mention is that each of these vertical lines is actually corresponds to one species. So these have all been ranked order by median size. So and the reason why they basically look like vertical lines is that it's individual points that just forms a line because there's so many of them. But yeah, so benthic, more abundant both in terms of species richness and individuals. And there's the large ones are mostly pelagic ones. There's also a lot of small stuff in the pelagic. So we saw kind of a bit more sort of modal distribution. And when we look at that as a violin plot, so here we have all the benthic ones and the pelagic ones, and we flipped it on the side to make it more analogous to a trophic pyramid. See, all the small ones are pelagic, and then you get up on all the big ones are pelagic. And then you have the intermediate size classes. So if you're looking at this in terms of trophic pyramids, you get kind of a broad, dumpy benthic pyramid as opposed to a steep pelagic one. And these patterns hold when you look across biogeography. Here we've kind of grouped that data set into absolute latitude bands. So this is kind of what we saw in the temperate area and then towards the tropics. So this is all the data we had in tropics and then sort of moving up towards the poles. And consistently, really, you have a steeper benthic size spectra. So that's what you're seeing here. And whereas the pelagic one is shallower. So that means that it's more dominated by large individuals. Because those, we have these two modes, so small individuals more abundant in pelagic, and large individuals more abundant in pelagics. And again, this single dome that we're seeing in the benthic is the size classes which are benefiting from all the hiding holes, you know, all the nooks and crannies, to use a good English term, on the seabed. So next, in order to explore the difference in vulnerabilities, we wanted to go into each individual location and then look at the influence of human pressures accounting for different environmental variabilities. So to do that, we extracted 3 size-based indicators. So we had small fishes, the typical size of this first mode and then the typical size of the large fishes, and then the slope of the size spectra. And together, these gives us basically different sort of architecture of what the size structure at an individual location would look like. And these partial plots here is, once we account for all the environmental variability at the global scale, this is what you're left with. So here on the x-axis, you have travel time to market on the 3 different protection levels. So highly protected, so no take, no fishing allowed, partly protected, and open to fishing. And on the y-axis, you have our large fishes. So these are our sharks, our predators, our rays, our tunas, and then our small fish sizes here. So again, it's quite a lot going on. But in general, we're seeing that fish becomes larger the further they are away from markets. So that's kind of what we expected. So each of these regressions are positive. So the further away you are from markets, the larger fishes get. And this happens to both relatively small fishes and relatively large fishes. Okay, so it's consistent across size classes. However, the biggest response we're seeing in the pelagic. And this here is so really remote, but in highly protected areas, is the only area we're seeing large size classes. They're completely absent in other groups. And this effect of remoteness, it becomes more pronounced for pelagics. So you can see that the slope becomes more pronounced the stricter your protection is. Whereas for benthic, it's the other way around. So you're seeing here, you get less of a signal with remoteness under stricter protection levels. Which means that your biggest relative change is actually close to markets for benthic protection. Yeah? Does that make sense? And these, as an aside, these protection categories are according to the IUCN, MPA categories. And when we're looking at the size spectra slope, you really get to see, how sensitive pelagic size structure are. You get a complete, so here you have size spectra, slope values, so, minus 0.1 and minus 2, and you get the full sort of change in the size structure is happening in highly protected, under remoteness. So that's basically the only way, really, to get those pristine size structures. What does this mean in terms of trophic pyramid terms? Well, for pelagic, you get a strong steepening. And another thing I should have mentioned is that for the benthic one, you actually get a shallowing of the slope. This isn't particularly significant, but it is interesting that it's consistent across protection status. So what that means is that for your pelagics, you get, looking at this in terms of trophic pyramids, you're getting a very much a steepening. You're getting a small dumpy pyramid, whereas for your benthic one, you're also getting a dumpy pyramid. So you're having a kind of a convergence between your trophic pyramids in both pelagic and benthic systems. And they're kind of converging towards this new artificial structure, which is less stereotypical than you would find in pristine systems. And I admit, to me, it was a bit counterintuitive when I first saw this. I get what would happen in the pelagic. You are seeing a loss of those large size classes, the sharks, the tunas, and so on. But I wasn't really expecting to see this kind of shallowing of the benthic one. But turns out this is actually quite common. Here's data from Coral Reef by Graham and friends. And here you can see their sort of signal of their trophic pyramid as the increasing pressure. So here is kind of high pressure, low biomass, and then sort of this is pristine. And what you're seeing is actually a slight, if you're looking at the average trophic level, it's actually increasing the lower the biomass level is. And this is because, in benthic systems, you have a lot of compensatory mechanisms. There's more species, there's more functional redundancy. And I'm sure you can think of examples of this happening, but if you're going to, for example, the Caribbean, and you see sharks are being removed, but then suddenly you have a lot of moray eels, which are sort of taking the role of sharks. And you're seeing the same thing in a lot of benthic systems. So there is, to some degree, there is more redundancy in benthic systems than in pelagics. Whereas in pelagic, if you're removing the big sharks, there's no backups. You're removing the big sharks and the big tunas, and there's just not enough species to sort of take that place. Another thing that could be at play is that benthic systems under humid pressure typically lose their habitat complexity. Here, looking at coral reefs, and when that happens, and I guess this is counterintuitive, but the average size of the predator actually increases in benthic systems. And that's because you remove those refuges, you remove all the sort of hidey holes, and there's more direct energy transfer between your herbivores and your predator. So even though there's lower overall levels of biomass in fish, your typical size of your predator actually increases. And that really is, it's actually getting some of the attributes of pelagic systems. When you are flattening a benthic system, it actually becomes more similar to a pelagic system. And this kind of might be explaining what's going on here. So what are the policy application of this? Well, the good news is that pelagic MPAs, they work. And they work if they're remote and highly protected. For benthic MPAs, they actually are the most effective near markets. So that's good news as well, because it means that there's huge potential for effective conservation, for nice gains, as long as you protect that habitat structure, which benthic MPAs are very good at. It does mean, however, that we need to be cautious about the expansion in pelagic fisheries, because that threatens that top-down control, leads to the sort of collapse of the size structure. So we really want to caution against that. The other thing is that this is the state of things now, but it could change in the future. So it could be that, perhaps it will become worth it for fisheries, for industrial fishing fleet, to go really remotely to catch the large benthic fish. At the moment, the most pelagic fisheries tend to be more wide-ranging, but that could change as well. So we might start seeing the same signal applying in benthic ecosystems. So I just want to plant a flag there that I'm not saying that this means that go catch all the last benthic species. In terms of the impact of this study, it's mostly, so far at least what I've heard was mostly positive. We did get some pushback at C4 source. We were told, I think we were lumped in with one of Mark's papers as well. And for me, this was I don't know, I took it as a bit of a badge of honor, really, because I felt a bit like I was being chosen by Voldemort, but anyway. Briefly, I want to talk a little bit about where we're going to go with this next. I mentioned that we've been working a lot in Chagos. And now I want to see, we've talked about the particular circumstances where pelagic MPAs are effective. Now I want to see how resilient they are. We saw a massive spike in illegal fishing during the pandemic. We've done follow-up surveys with our bruvs, so we've gone back and we've sort of seen, well, what's left in terms of the fish. The good news is that this illegal fishing has actually tapered back down, so it's actually returned to pre-pandemic levels. And that was due to sort of quite interesting novel bilateral agreements with India. So these vessels came primarily from India as opposed to Sri Lanka, which we used to be the dominant source of the vessels prior to the pandemic. So we now want to look at what are the sort of once the dust has settled, what's what's the ecological impact, and what were the changes in the social driver for those fishing fleets that led from that sort of changing dynamics because of that social ecological shock that we all felt during the pandemic. Another area, so now that we've sort of identified that kind of divergence in sensitivity between pelagic and benthic system, is whether that leads to a decoupling of pelagic and benthic food webs. So this is a proposal at the development stage. I've tried to find potential partners this week, so I appreciate those of you who listen to me pitch to you. And the idea would be to sort of combine various data sources, longitudinal studies, some of the historical data, combining both pelagic and benthic biomass and see how they co-vary over time and space. Finally, I want to stress the collaborative nature of this work. As I mentioned, this BRAV sampling program is obviously a large international collaboration. The field component's been spearheaded by Jessica's team at Jessica Mewig at UWA, with input from lots of partners. I forgot to mention this is a MARHAB output as well. And finally, acknowledgement to the insane effort involved in analyzing all this video footage. So technicians, PhD students, and I'm told, I've done a little bit of this, but not so much. I'm told that the pelagic BRAVs are particularly strenuous to analyze because they're constantly bobbing up and down. So you can be in your office and you actually get seasick looking at these things. And finally, money. Thanks very much.
Thank you, Tom, for this excellent presentation. Questions straight away.
You already have asked a question about this. So my main question is that these size trends, I'm assuming they are like community level, right? So the reason why sizes are getting bigger or smaller for pelagic or benthic species could be either because at the species level they are getting bigger, or because some species are getting replaced by bigger species as species protection. So which of these factors is more important?
Yeah, so the question is it the different species composition or is it different body sizes within the species composition? In this case, it's both. You see widely different species, and you see some species in both. You definitely do. Some will have some jacks will have pelagic larval stages, so you see the tiny one in the pelagic, and then you see the sort of the adolescent and the adult stages on the reef. That seems to be the case for a lot of species, but then the larger size classes, the pelagic ones, tend to be uniquely pelagic. I didn't go into the details, but here we define pelagic versus benthic not on account of that's purely empirical, so it's just where we saw the animal, rather than just sort of looking at the taxa and say, okay, it's primarily pelagic. That's not how we defined it. It was just purely empirical based upon our sampling.
Stuart from JMCC in the UK. Thanks for that. Really interesting results, particularly the benthic MPA effectiveness.
Yeah.
And I just wondered whether you looked at or considered looking at the gravity model from Joshua Sinner, which incorporates the distance marking and also population size.
Yeah. So the paper from Joshua Sinner, is one where they've. Yeah, so they basically have these columns, and then assuming that is that you're going to have a more pronounced pressure near these big cities. Sure, I mean, that would have given us a bit more power, but the bulk of it is, there is this particular threshold that happens, and I talked about it briefly, but the moment you have the infrastructure required to support fishing vessels, the way you see outside our window here, there's a massive threshold that happens in terms of the impact. So, given the already quite busy model, you know, there's a lot of variables in here. We sort of opted for some shorthands. But you're right. That you probably would have detected one stronger signal if we had done that. Yeah.
We've got one more question. I think there's one back here.
It's a great job, and even better than I expected. I love it when you can put pattern install this huge amount of data. So I'm also struck by how sensitive the pelagic is to the overfishing.
Yeah.
So the prevailing paradigm in MPAs is that you just stop off and throwing your solid river and fishing on the surface carries on as normal. So what do you think of this idea of separating pelagic and benthic in MPAs?
So what do I think about this idea? So do you mean now from a
Allowing pelagic fishing to go ahead in MPAs instead of holding these areas protected?
Yeah. I mean, here we had data, both pelagic and benthic data from the same sites. You know, so it was treated differently from a sampling method point of view, but not from a policy point of view. I don't know if that answers your question.
Should the policymakers be separating pelagic and benthic conservation?
Right. Okay, yeah, that's, yeah. So I need to get funding for this follow-up study. And no, but I think it's a very good question. I think it's unrealistic to expect much in terms of much pelagic conservation to be achieved by small coastal MPAs. I think that is what we're able to do, on the coastline. If we were to be able to put something like a Chagos-size MPA, but that realistically isn't gonna happen because of because of the stakeholders. And so to me the question is, how much benefit do small benthic MPAs or small, how much of those benthic conservation gains, how much in terms of pelagic productivity is boosted by virtue of having these small high-quality habitat protected along the coast, and that's kind of my next, big question: is does that yield, small pit stops where big pelagic predators can sort of nip on the top of benthic food webs? I think that's a really interesting question, but it's still a question I want to answer.
Thank you.